Hello, everyone. If you're new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. Today's topic will be discussing how to establish the competitive landscape within the Scientific Validity Report. Our presenter today is Dr. Sarah Chavez, Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team.
Okay, thank you, Olivia, and thank you to everyone who's joining us today. During today's session, I would like to review the scope and the purpose behind the Scientific Validity Report, which I tend to abbreviate as the SVR. In particular, I'd like to discuss how the SVR helps you to establish that your IVD can be considered state-of-the-art. So my goal today is that by the end of this presentation, you will all have a better understanding of why the SVR must include a clear discussion of state-of-the-art, where to identify relevant performance objectives for your IVD, what data can and should be included in your competitive landscape, when to include detailed screening and appraisals of the articles identified, and finally, how to present the data in order to determine the acceptable range. Now, SVRs will certainly vary, and they should be customized based on the unique needs of your IVD. However, IVDR regulations are quite specific in defining what they mean by scientific validity, and that definition is that scientific validity is the association of an analyte with a clinical condition or a physiological state. So I think it's helpful to keep that definition in mind as you develop the outline for your SVR. There are some important references and feeder documents, such as your IFU, that will help you get started. But as we will be discussing today, the Scientific Validity Report will rely heavily on defining the state of the art. So that's where I think having experience with medical devices and MDR can be extremely helpful. The SVR is a new requirement for all of us, and the approach that we'll take to establish state-of-the-art in IVDR is essentially the same approach you would take if you were writing the SOA section for a medical device in a CER. The first thing to point out is that if this is your first time planning an SOA section, expect to conduct multiple systematic literature reviews. This is not just a quick PubMed search. It's a very detailed, organized, and very well scrutinized process. So please don't try to rush it. You want to go into this process with a very clear plan and a solid understanding of what the notified body expects to see as a part of your final report. The articles retrieved in this first systematic literature review will provide the information that's relevant to the background of the state-of-the-art section. So that will include things like the medical condition, the population, as well as diagnostic alternatives and the risks and benefits associated with clinical testing in general. So we discussed this first part of the SOA in previous webinars, so I encourage you to check out that webinar if you've missed it. Now, today we are focusing on the second systematic literature review, and that will support your competitive landscape. In MDR, this section is sometimes referred to as the maturity of the technology section, because really this is where you go into detail about the IVDs that can be considered the most similar to your IVD. This data will be appraised and extracted to present your competitive landscape. So you should have a solid sense of not only your competitors, but the measures by which you can compare the performance of these competitors. In today's webinar, we will be walking you through the SOA process from start to finish, and I hope to show you how the two independent searches will ultimately help to tell the story of your IVD in the context of scientific validity. But before we get ahead of ourselves, let's take a step back and think about the purpose of the Scientific Validity Report. So first and foremost, are we all confident that we understand why establishing scientific validity requires defining what we mean by state of the art? Generally speaking, the state of the art can be considered what is currently and generally accepted as good practice in technology and medicine. So according to our source document, MedDev 2.7 Rev 4, state of the art describes what is currently and generally considered standard of care or best practice for a medical condition or treatment for which the device is used. If we were to apply the same definition to IVDR, we can think about the state of the art as what is generally considered to be the standard of care for the diagnosis of a medical condition or a physiological state. The state of the art does not necessarily imply that you are the most technologically advanced solution for diagnostics, but it should include a discussion of the medical condition, alternative diagnostic technologies, clinical practice guidelines and recommendations, and the risks and benefits associated with using this type of test in order to make a diagnosis. And in addition, the scientific validity report is expected to explain the association between the analyte you are detecting and the medical condition it's being used for. These are all critical sections within a typical SOA, and that information will be gathered in large part from the articles that you've retrieved through your state-of-the-art systematic literature review. So you will be including current recommendations and guidelines related to clinical diagnostic testing. That will likely include your SOA articles, but it could also require some ad hoc searches in order to identify statements that have been published by experts and organizations. This can be fairly straightforward if your analyte has a very well-established association with that particular clinical condition, or if it has a long publication history. But suppose your IVD is designed to detect a novel analyte, or if there's still limited evidence regarding your analyte in published literature. In that case, you are still expected to present alternatives and discuss current clinical guidelines. But you need to carefully consider the strategy necessary to establish scientific validity. This is why we recommend that you begin your scoping process very, very early in the PER preparations, because limited data supporting the scientific validity report can and will significantly impact the quality of the PER as a whole. And now finally, you will need to define your competitive landscape. Competitors are not the same thing as alternatives, so keep that in mind. This section is where you will establish the safety and performance measures as well as your acceptance criteria using the data that you have extracted from many articles that you've retrieved through your competitor searches. The goal is to compare your IVD to your closest competitor IVDs, and then you will use the PER to show that your IVD performs as intended and meets these pre-established acceptance criteria. So be prepared to document each of these searches in detail, and you should be expecting that you need to present your results very clearly. The process of building the SVR template relies heavily on the results of your systematic literature reviews. But the process itself must also be objective, reproducible, and well documented. The notified body will expect to see that relevant articles were identified, screened, and appraised in a very transparent, clearly defined manner. There are two different systematic literature reviews here that we've discussed so far, and I will just provide additional details later in the presentation regarding those two sources. The information contained within the SOA is critical to not only the SVR, but it will directly impact your CPR, the clinical performance report, and the APR, the analytical performance report. So this really illustrates the need for the SOA section to connect each of these reports together to ultimately tell the full story of your IVD. Okay, so now we know that we need the SOA to provide that background and the context for the clinical condition, but also to set the standards by which we can measure our IVD relative to the competitive landscape. So now what? Where do you start? We need to know clear safety and performance objectives, but how do we ensure that we have selected the best ones for our particular IVD? Based on our experience writing CERs, a good place to start is by clearly defining your intended clinical benefits. These are not just commonly reported endpoints. And in our experience, that seems to be one of the major points of confusion for IVDR clients. Clinical benefits can be informed by guidelines, recommendations, and medical standards, but they should also be meaningful within your own quality management systems. While direct clinical benefits should be supported by clinical data, indirect clinical benefits can also be demonstrated using other evidence, such as preclinical or bench testing data, things like compliance to product standards or common specifications. This could include real-world data, including registries or information derived from insurance database records, et cetera. Or it could include data from another IVD that's used with your subject device, which does not have direct clinical data. For example, in the MDR world, we often will collect data from a stent that can be used to justify the safety and performance of an associated guide wire. Now, what are your intended clinical benefits? And just importantly, what are the reported risks which will be included within your risk management files? This is where the multiple moving pieces and reports should start to become clear. First, you should take a look at your IFU and other feeder documents. That should help you determine your intended clinical benefits. The need to clearly define those is a new requirement for IVDR. Do you know what your clinical benefits are? And if not, where do you start? This is where the clinical practice guidelines and the diagnostic algorithms and standards are incredibly critical. So where do you find these recommendations? A well-crafted systematic literature review for your SOA section is a good place to start. The SOA search often includes mesh terminology that will help you capture relevant clinical practice guidelines. And while ad hoc searches may still be necessary, the systematic literature review will provide you a good start. Until we really start to receive feedback from notified bodies and get a more complete picture of the expectations for each of these reports, we're relying on our extensive experience with writing CERs for our medical devices. The Performance Evaluation Report, or the PER, will verify safety and performance of the IVD, which includes a detailed assessment of the clinical benefits of the IVD when it is used as intended. To accomplish this goal, we must define the state of the art and explain how this particular IVD technology compares to similar technologies within the entire diagnostic landscape. So in addition to establishing that this assay is a scientifically valid approach for making a particular diagnosis, we must also show how similar IVDs perform. So that's really the purpose of the SVR. You will also need to clearly define the intended clinical benefits of this diagnostic tool in the context of the patient's clinical course. That's where the CPR will come in. So together, the scientific validity report and the clinical performance report will seek to define and justify the safety and performance measures by which you will judge your IVD against similar competitor devices. The competitor data lives within the SVR, and so it is going to be a critical input for many of these other reports, including your post-market performance and surveillance plans, because it justifies the choice of safety and performance objectives, but it also establishes what may be considered acceptable safety and performance for these measures. One aspect of the SOA section that comes as a surprise to many of our clients is the data that we are presenting here in the SVR is not actually the data on your IVD. The SVR is establishing the competitive landscape. So you should expect to present a thorough assessment of the performance of other IVDs, not your own. But hopefully this schematic will help you see the complementary nature of each of these reports and illustrate the need to carefully choose and select those performance objectives very, very early in your IVDR writing process. The objectives will be discussed and justified at multiple stages in the PER. So if you decide late in the process to change one of these safety and performance objectives, It could have a ripple effect, and it will inevitably lead to rewrites, edits, or delays. Organization is critical to keeping your PER preparations on track. And the SVR is really a solid roadmap that will help you identify gaps in your objectives before they have a chance to carry through to other aspects of the project. So on that note, I'd like to discuss the next stage, which is that competitive landscape. As you develop the SVR template, think about things like not only the type of data you're collecting, but also how are you going to connect that information from the background section to support and justify the choice of the safety and performance objectives you're presenting. The SOA and the competitor searches are separate and each have distinctive purposes, but they should be complementary in order to support your choice of performance objectives. You need to identify data that validates the overall scientific validity of your IVD technology and also supports your assertion that the subject IVD is capable of providing an accurate, state-of-the-art clinical diagnosis based on your analysis of the competitive landscape. So the easiest way to approach this is to think of the SOA section as a funnel. The SOA section within the scientific validity report should show not only how your IVD fits into the entire clinical diagnostic landscape, but it should also provide a robust and an unbiased assessment of how your competitors perform using well-defined, carefully selected objectives. The data from this section will establish your acceptance criteria by which you will assess your own subject device performance within the scope of the competitive landscape. And this is how you will establish that your IVD may be considered state-of-the-art within your final report, the PER. So as you develop your SVR templates, think about how you will begin to connect the information from each of these sections in order to ultimately justify your choice of performance objectives. These are two different searches with distinctive purposes, but they should be complementary. And the overall scientific validity of your IVD technology should provide an accurate, state-of-the-art clinical diagnosis. The competitor device systematic literature review will include carefully crafted search terms and filters because the articles that you have retrieved through this search should be carefully and methodically screened. You will define clear inclusion and exclusion criteria, and then you will be expected to document that the articles you have included or excluded during this entire process were assessed and appraised based on the same objective and unbiased standards. The screening and appraisal process must be reproducible, and it should be justified within the SVR to establish that you have presented a robust, unbiased approach in order to identify articles with clinically relevant performance data. The process of systematic literature review has long been the gold standard methodology for evidence-based research. It's been used in academic and professional arenas to help develop clinical practice guidelines and support academic-based research initiatives. So at its core, the purpose of the systematic literature review is to ensure that you have identified relevant scientific data through a process. So this is a good time to point out that this is not optional. Systematic literature review is mandated as best practice, and it can also be an extremely daunting process. With the help of one of our qualified reference librarians, we use systematic literature review to design our search using proper mesh terminology and a robust search platform that allows us to search across multiple databases. This strategy is very critical to the next stage, which is screening, because each of the articles that you identify in your search should be screened for relevance based on pre-established inclusion and exclusion criteria. If you are stuck with a poorly designed search, that could easily result in thousands of articles being retrieved, which could mean that you should expect to spend a significant amount of time unnecessarily during that screening step. The data extraction is the stage in the process in which you've identified articles with potentially relevant data related to your pre-established performance criteria. And in the case of many IVDs, the performance criteria may encompass data such as sensitivity, specificity, false positives, false negatives, or even limit of detection. And then finally, we should present the data that has been extracted, but also present the process by which we undertook the systematic literature review process in order to arrive at this data set. And that brings us to the final step, presenting your data. How do you plan to extract, assemble, present, and synthesize the data that you have collected to ultimately arrive at your goal, which is acceptability criteria for your subject IVD? I'd like to use an example of a data set that could be extracted for a typical IVD. And here I've presented it in a tabular format. So let's suppose your IVD is a test for Giardia, which is an intestinal parasite responsible for 200 million cases of infectious diarrhea annually. In this example, there are multiple types of diagnostic tests that can be used to make a clinical diagnosis of Giardia infection. Direct stool examination involves looking for cysts or parasites directly. And while it has excellent specificity, the sensitivity is quite low. And in contrast, the sensitivity of an antigen-based test or a molecular assay are much higher, but with lower specificity. Similarly, the false positives may reflect on the specificity of a particular test. A test with a high specificity would be expected to generate relatively few false positives. And conversely, a test with a high sensitivity would be able to diagnose the majority of infections, which should result in fewer false negatives. Now, suppose your test is an antigen-based test. In this case, the SOA section would present your alternatives, which would be the results of direct examination or molecular assays. Those tests can be presented as alternatives, but clearly they provide much different ranges compared to the antigen tests. So keep that in mind as you're defining your competitors versus alternatives. If you don't select the appropriate competitors, or if you confuse an alternative for a competitor, that could dramatically affect your acceptance criteria. Now, when determining your acceptance criteria, the lower range of performance measures reflects the bar that your data must exceed. So in this case, antigen detection has a sensitivity of 44 to 100% amongst all of the competitor antigen tests and the data that we've collected. So when you identify the data within your CPR, your IVD should perform better than the lower end of this range. That is to say, you should perform with a sensitivity of at least 44%. For a safety objective, your range reflects the top end of what should be considered acceptable and state-of-the-art. And so you want to show that your device is better than the least safe option. So again, in this case, if the rates of false positives are 10 to 15%, you would want to establish that for your device to be considered state-of-the-art, your rate of false positivity should be no more than that top end of 15%. So as you can see here, this data shows that in order to present an acceptable risk-benefit profile, your IVD's performance should exceed a sensitivity of 44%. But had you selected a molecular assay as a competitor rather than as an alternative, now you've raised your bar to at least 65%, as molecular tests have higher sensitivity than a typical antigen-based test. You could also inadvertently lower the bar in terms of your safety objectives if you had included molecular assays as competitors, meaning that instead of the appropriate acceptance criteria of less than 15%, you would instead present a rate of false positives of no more than 6%. So the purpose of this table is really to hopefully illustrate that choosing an inappropriate competitor as opposed to an alternative could raise or lower that bar, which could make it difficult to meet the criteria within the data presented in your CPR. So be prepared to identify and select the most appropriate and directly comparable competitors in your SVR. So I hope in today's session, we've addressed some of the most commonly received questions regarding how to begin the SVR process. We hope you have a better understanding of why the SVR must include a clear discussion of what can be considered state-of-the-art, where to identify those relevant performance objectives for your IVD, what data can and should be included in the competitive landscape as opposed to the alternatives, when to include detailed screening and appraisals of the articles you've identified, and ultimately how to present the data in order to establish your acceptable range.
All right, thank you, Sarah. That was a great presentation. I think you really laid out those steps clearly. So now we have a big chunk of time to start answering some questions. So if you haven't sent your question in yet, go ahead and use the Q&A button on your Zoom menu bar. We'll try to get to as many as we can. I want to remind everyone here today that you can also reach out to us at consult@criterionedge.com to learn more about our services or schedule some time to talk to our team. We've recently worked on several SVR and PER requests, and we'd love to chat with you about your current project needs. All right, Sarah, let's get to our first question. All right, our first question today is, how can we start to perform the scientific validity process?
We get this question a lot. As we're beginning the process of helping a client with preparing something like a scientific validity report, we start with a scoping checklist. It's important to know what inputs you already have available to you, what decisions have been made up front, and basically just make sure that you are well aligning all of your internal documents. So I would say your first step is really to get a good sense of what your intended purpose is, what the potential clinical benefits would be for your particular test, who your patient population is, and really start to drill down on the terms and the mesh terminology that you would use to begin your PICO searches. So your patient population, the indications, the comparators, and the outcomes. That's really the best place to start. Figure out what you already have available to you, and then start to craft some of those early SOA searches.
All right, great, thanks. Here's our next question. Is a competitor supposed to be commercialized?
So in our experience, it often is. And in particular, like for instance, in the COVID testing arena, it's they're recommending that as you select a competitor, it should actually be CE marked. But generally speaking, competitors are going to be something that you want to be able to collect data on and make sure that that data is standardized. So we often use these commercialized competitors in order to ensure that these are not in-house assays that you're comparing from one hospital to the next. There could be quite a bit more variability if a competitor is not commercialized. It's not to say if there aren't any assays available that are commercial assays, that you can't use that data, but that's certainly a limitation of it when you are dealing with variable in-house assays.
All right. David is asking if you could please repeat the connections between sensitivity and specificity to false positives and negatives.
So yes, so we find that in our experience, we've been collecting data on both because ultimately the sensitivity goes to and specificity go to the performance of the device. But from the patient's perspective, a false positive and a false negative would directly impact their potential clinical care. It could affect how they're going to receive care if they get an incorrect result. So really the sensitivity and specificity are kind of those analytical performance measures, whereas the false positives and false negatives are directly related to the clinical safety of this particular assay. So they're definitely related though. So a sensitive test means that it's most likely going to detect the vast number of positive cases. So if you're highly sensitive, you should get very, very few false negatives. And if you're highly specific, you should get fewer false positives, because if you have a high degree of specificity, it means that there's maybe limited cross-reactivity or a potential for you to pick up on something accidentally that could result in a false positive test result. You may still encounter case reports that could report on the potential for a false positive or a false negative result. Those wouldn't necessarily be collected as part of your landscape analysis, because often these case reports are an N of 1, but they can still help you identify those risks and clearly define why, if you have a high rate of false positives or false negatives for your test, what might that actually, what are the causes for that? Are there particular limitations of this type of assay as a general rule? So they both will, we present both of them because really that scientific validity report, our role in writing that report is to help link together the data on your device that you're presenting in the analytical performance report and the clinical performance report. So this is where your comparator data is going to live, is within the SVR.
Mm-hmm. Great answer. I hope that helped answer your question, David. Getting on to our next question. This person is asking, do we have to capture all competitor products' data?
It depends on what you mean by all data. Depending on what type of assay you're writing for, there could be hundreds of competitors out there. You should not expect to collect data on all 100 competitors. You would have tables that would go on for miles. Instead, we recommend that you select some competitors that seem to be the most widely used, the most frequently used. If there are particular competitors that are often used as sort of like that benchmark, that standard by which other newer devices are measured up against, those would be the types of competitors that we would try to make sure that we include in our list. You can often work with your marketing division and determine who they would consider to be your closest competitors within the landscape. You can limit it to competitors that are already CE marked or who are used widely in the European market. But if you're talking about data in terms of the data that's retrieved from a systematic literature review, then yes, you would want to say, if there are 100 tests on the market and I've decided to focus on 5 of them, I will want to capture all clinical data presented for those 5 competitors that I've identified through systematic literature review. You wouldn't want to exclude a particular data set that seems to be sort of an outlier for the rest of them. You would want, or if you do identify an outlier, like maybe your particular test is being used in a different patient population, be prepared to justify that as part of your exclusion criteria. For instance, if you have a test that is used in the general population versus in an immunocompromised population, there may be issues with sensitivity or specificity of that assay, or if it's used in an adult versus a pediatric population, those may be differences. So it will align with your IFU as far as which data you're going to capture to make sure that it's part of your own intended use and the purpose of your device. But yeah, in terms of the competitors, you do not have to necessarily describe data for every single possible competitor out there.
All right, great. Thank you. So this next question, this person is asking, if the state-of-the-art systematic literature search is carried out after the initial drafting of the performance evaluation plan, is it expected that you loop back to the performance evaluation plan and update it? What are your thoughts on this, Sarah?
Yeah, so I think this is a really important point, that this whole process of writing, it's not looking for the end goal of creating a report and then you're done. It's a process. So it is iterative. It is going to loop back and feed back on itself. So as you've conducted a systematic literature review, suppose you've identified a performance objective that literally everyone else in the field seems to be collecting, but for whatever reason, your initial test was not collecting that same data. That can inform the future collection of data through your performance evaluation plan. So you may say that, you know, up until now, we haven't been collecting data on false positives, but now we're recognizing that in the literature, some folks have noticed that there's a particular cross-reactivity. So now we're going to start collecting false positives as part of this new information that we've received as part of our state-of-the-art search. That shows the notified body that you are using the information that is newly collected to inform your processes and systems, that it's not just, you know, we've always done it this way, and these are the objectives, and we're never going to collect anything other than these. So yes, so be prepared that if you do identify some new information, that it could directly impact some of these other key documents.
Definitely. Our next question, Sarah, I think is one that we get somewhat often, but this person's asking, how far back do we have to go when finding references?
This will definitely depend on your test, in my experience. If you have a test that is sort of the tried and true, this is like it has a publication record going back 20 years, you wouldn't want to include data going back to all 20 years. We, as a rule, pick the previous 5-year period. And then as you're doing updates, you can collect data on that previous 5-year range. We have found that for some devices where they are these sort of legacies, you know, they've been around for so long, you may not have a whole lot of data presented in the past 5 years. So in that case, you can also rely on data from systematic literature reviews and meta-analyses that have gone back through all of those historic data and provided those ranges for you. But yeah, as a rule, I would say for your competitive landscape, 5 years is pretty standard. State of the art may go back a little bit longer, depending on how much difficulty you're having with finding some of those background articles.
All right. Our next question comes from Sylvia. Should competitor data also be used to establish analytical performance acceptability criteria?
Yes, so I hope that sensitivity, that's why I presented both the sensitivity, specificity, false positive, false negative. I left off some of the other analytical performance criteria, but we also collect things like area under the curve or odds ratios, hazard ratios, you know, things that would be limit of detection or cutoffs. Those are other criteria that we would also collect as part of the scientific validity report in order for you to, again, to compare your data that you're collecting as part of your APR to the data set for your closest competitors. So yes, that SVR is going to directly feed back and loop back to your APR and your CPR.
All right, great. Thanks, Sarah. Here's our next question. What is the acceptable minimum number of papers to be analyzed in the scientific validity report?
This is sort of that magic number question that we get a lot. When you're dealing with the SVR and these systematic literature reviews, there really isn't a magical minimal number. I would say that as you conduct your search, if you're noticing that you are not getting any data, that's an issue, and you should probably reevaluate your search strategy to see if maybe you've been too restrictive, or if you've selected the wrong search terminology to properly capture all of that data, or perhaps you haven't identified a really critical competitor. In that case, I would often go back to the clinical practice guidelines. Those are really going to be a helpful source for identifying some of those sort of standards which within the field as a whole. But yeah, there really isn't a minimal number of articles or studies that you should include. But if you do have a very low number, be prepared to show and justify your approach for the systematic literature review and show that you tried your best, that maybe this is just a technology where there's not a whole lot of data out there yet. Or if it is an older technology and nothing has been published in the past 5 years, presenting data on meta-analyses of these historic articles could be another approach to take to sort of help to supplement that.
Thanks, Sarah. I also want to mention that if you have any specific questions about your device, you're welcome to book a free 30-minute consultation. We're happy to have a chat with you about your project needs, talk to you a little bit more about any of your, more specific questions you have about your device. And you can book an appointment with us from the link directly from the chat. So Sarah, our next question, this person is struggling with how the different reports fit into the PER. So do you have any guidance on how to compile all these reports?
The way I look at these 3 reports is that they're all supposed to be very complementary. The clinical performance report and the analytical performance report will present your data. That is what is going on with your particular test. The APR you can kind of think of that as more sort of that in-house data. If it's under ideal circumstances, sort of those preclinical data sets that you collect in a laboratory setting, in-house, how well can it perform? How well is it intended to perform? Are there any reasons why the analytical performance might not be meet expectations? That's the APR. The clinical performance report is really, once your device leaves the walls of your building, what are people doing with it in a clinical setting? How accurate is it as far as providing that diagnostic information? That's where things like the false positives and false negatives, those types of data sets will come into play. It will often be presented as sensitivity and specificity, but really the main difference, whereas things like sensitivity and specificity can also be analytical performance measures, the clinical performance is really how does it perform in a real world setting, under like when it's put to the test in real live patients, how does it perform? So you can see that like under ideal circumstances, you have a limit of detection of a certain amount. And once it's brought out into the real world and samples are left on tabletops for extended periods of time, or, there's individual variability, you don't have known standards of what is positive or negative, that's the clinical performance data. And ultimately, the SVR is going to tell you how others who are close competitors of yours perform under the same circumstances. When they're in ideal laboratory settings, what is their expected sensitivity, specificity, false positive, false negative, so that when you collect your own data, you can show how well you compare to your closest competitors. So they're definitely complementary, but your data lives within the APR and the CPR and the competitive landscape, as well as the justification for your overall approach that you've taken, and the data that you're collecting lives within the SVR.
All right. Moving on to our next question. What processes can be used to define acceptance criteria? Any thoughts on this?
Yes, so this is something that I think a lot of folks go into the process with kind of a little bit of confusion about the differences between performance measures or performance outcomes and acceptance criteria. As you're collecting the data from those competitors, you're going to identify ultimately a range. So you may have one competitor reports a sensitivity of 99% in one assay, and the next competitor data set that you collect reports a sensitivity of 90%. Well, your acceptance criteria is going to report that overall range. So of all of the articles that I identified with competitor data, I have a range of sensitivity from anywhere from 90 to 99%. My acceptance criteria is then based on either the upper or lower bounds of those ranges, depending on whether you're dealing with a safety or a performance measure. So as you're developing the processes, you should expect to clearly present all of the data for your competitors in a tabular format often, that shows where that range was derived from, how many articles total did you include, are there any nuances between those articles, were they all in the same patient population? You may not want to ultimately pool data sets for adult versus pediatric, or for specific patient populations if those ranges are quite different. So for instance, in a lot of diagnostic tests, the sensitivity may be quite low in a pediatric population. So if you were to have pooled data set for your acceptance criteria, you may not actually, it may show a very, very wide range. So in that case, it may be helpful to define separate acceptance criteria for adult versus pediatric populations, depending on what your IFU states are your intended patient populations. So it's definitely, this is the part of the writing process that we definitely, as writers, we collect all the data. You don't really know what you're going to get until you actually collect it. And then by looking for sort of how those ranges appear within the data that you've collected, that's how you're going to justify your acceptance criteria, and how you're going to justify whether or not you need to slice those criteria into different subgroups, or if you're going to group them all together.
Nice. All right. Thank you, Sarah. Here's our next question. This person is asking if you have any advice on how to build an SVR for products for general laboratory use, which do not detect or measure analytes, but can be considered as accessories to an IVD medical device.
Yes. So I know this can be quite confusing. When you're Class A, the current understanding is that you often are not required to complete a CPR, a clinical performance report. But in terms of the SVR, you would still want to justify the use of your particular reagent in a clinical setting. So for instance, let's say you're comparing between different buffers that you're going to use for a particular test. You may be able to identify scenarios by which other types of buffers are used, or other assays in which your type of buffer is used. So you can still collect those data sets. There may be certain scenarios in which one buffer is preferred over another, maybe because of sensitivity. It affects the sensitivity, it affects the stability of a particular chemical in that sample. So we've encountered this for things like let's say you're doing something like a PBS buffer, like a phosphate buffered saline. If there are other alternatives to that, you would collect data showing that, sometimes these tests can be run using a different buffer, but PBS shows improved stability over a wider range of temperatures, or something to that effect. So it may wind up relying more heavily on alternatives as opposed to direct competitors. You wouldn't necessarily need to be expected to capture clinical data for competitor PBS buffers offered by different companies. But it would be the approach that I would take would be to use for if your particular general use reagent was not available, what else could they use, and why would that sort of backup plan be better or worse than using your particular chemical? That would be the main approach that I would take.
Great. Thanks for the advice, Sarah. Our next question is sort of a two-part question. First, how do you use data from competitive products with completely different technologies or type of products or treatment? And is it sufficient to restrict the competitor search to devices based on the same technology?
Yes. So in response to that second question about the restricting the competitor searches to devices based on the same technology, I would say absolutely. That is going to provide you the tightest range of in terms of determining your acceptance criteria. You're not always that lucky though. Like, let's say you have an assay where there aren't a lot, it's a new type of technology, and you're novel, you're using a different approach to detecting a particular analyte. If everyone else uses histology and is using antibodies against a particular analyte, and you've developed a PCR-based approach. You may not be able to find other competitors that also are using that same PCR-based approach. So in that case, you would still use your scientific validity report to justify the approach that you're taking to detect that particular analyte, that analyte is still a valid way of making a diagnosis, whether you detect it using antibodies or detect it using PCR. Your technology is still a valid approach to making a clinical diagnosis. But as you're collecting competitor products from different technologies, you may be limited to those types of data sets. And in that case, I would recommend extracting different ranges depending on the technology. This is going to be very common, I suspect, in IVDR clients who are working with different types of PCR-based technologies. There are so many different types of molecular assays. So we're very careful and conscious of, as we're extracting the data, to extract information about the platform on which these assays are run, the type of assay, the type of technology that's being used, so that you can determine the acceptability of each of those different types of assays independently. So depending on how new or how unique your particular IVD is, it may be necessary to somewhat blur the lines between who you would consider a competitor versus an alternative. You would want to ensure that you justify your choice of including them in order to establish your acceptance criteria by showing that there are no direct competitors, but based on the state of the art and the overall alternative landscape, this is why you took the approach that you did.
All right, our next question is an interesting one. How would you approach the scientific validity report for an IVD real-time PCR kit?
Yes, so we have worked on quite a few of these. In my experience, as I'm approaching an SVR for a PCR, your analyte in that case is the RNA or DNA, depending on what your template is. You want to justify the choice of your the gene that you're actually amplifying. So you will discuss basically do all of your competitors also include the same gene? Are you amplifying more than one gene target in order to reduce the chances of those false negatives? If you have something like a COVID test where there are concerns about the virus mutating, some of the newer PCR kits are employing multiple primer sets in order to ensure that if there's a discrepancy between one primer set resulting in a positive, one resulting in a negative, you can use that to help resolve and make an accurate diagnosis. Those are the types of things that you'll want to justify in the SVR for a PCR based approach. You want to justify your choice of genes, how many genes you're amplifying, where the primer sets are located, and whether or not this aligns with the same approach that your competitors are taking in terms of which particular gene are they amplifying, and how does that compare to any clinical guidelines that have been posted by reputable organizations? Do they all recommend using that particular gene in order to make that diagnosis?
All right. We have just a few more minutes left in our session today, so if you haven't sent us your question, you should do so now. We'll get to a few more. Here's our next one. How do you critically appraise literature in a systematic manner?
So we use a very set criteria where we basically, there are some standards that you can use for conducting these appraisals. We tend to present this as an appendix at the end of the scientific validity report, which will make it far easier to integrate it into the PER as a whole. But we basically established the level of evidence. You want to show that the data that you have included in your competitive landscape all are high quality, high level of evidence articles. This will take into account things like the total number of patients, whether it was used in the correct population as intended, whether it was used for off-label purposes, whether the type of study, if it was a randomized control study versus a prospective or a retrospective analysis, if it was a systematic literature review and meta-analysis compared to a single study. All of those things should be taken into account to provide you with a level of evidence. And then by presenting that in the appendix, you can ensure that all of the articles you've included in your current analysis meet a certain level of rigor, that they're high quality articles. Or if you are quite limited in the data that's being presented, you have now a way of explaining that as part of your review, that, you know, we did see quite a range, and it's because there is limited data in the published in publications.
Great, thanks, Sarah. Here's our next question. How can scientific validity be achieved when there is not enough data?
Yes, this is definitely a concern for a lot of our clients. Just because this clinical performance is a brand new requirement for IVDR. You know, up until now, once that you do all of your testing inside of your house, and that's your APR data, we've all had that. We know that our device performs under laboratory scenarios because we've tested it before it went out the door. But the scientific validity is going to take that a step further and say when it's being used in actual clinical real world settings. You often have to rely on the kindness of strangers that have used your device in their own studies and have published data on your device. If there is not a lot of published data, you may need to get a little bit more creative with how you identify data sources. You may have to look at insurance records databases, or you know, go into some of these sort of publicly held databases and registries in order to capture data. In worst case scenario, you may actually have to perform an in-house study in a clinical real world setting in order to collect that data. So I would say start by making sure that you have captured a wide net, that you've actually captured data on your competitors. If you're struggling with finding data on your competitors, it could reflect that there is that the data is just not very well published, or it could reflect that you've selected the wrong competitors or the wrong search terms. So I would say that everyone is always concerned with not having enough data, but if you look hard enough and you've exhausted all sources of data, then you may be stuck designing a study in order to capture some of that missing data set.
Great answer, Sarah. We are out of time. Thank you so much, Sarah, for your presentation today. For more questions on the content we discussed today, or if you'd like to learn about our services, you can schedule a free appointment with one of our specialists. Our contact information is consult@criterionedge.com. Thank you everyone for attending today and for your wonderful questions. We hope this presentation helps you get one step closer to IVDR readiness. Take care, and I hope to see you at our next webinar.